Okay, we'll go live in another few seconds and I'll let you know once we are live. Okay, we are live. Keshav.
Hi, good evening, one and all. A warm welcome to everyone to today's Journal Club on behalf of Academy of Clinical Embryologists India and President Dr. Sujata Ramakrishnan. I once again welcome you all to today's Journal Club. I'm sure you all had a long, tiring day and got yourself a nice hot cup of coffee or tea because there's so much of interesting concepts that our speakers going to really add on. And at the end, we're going to have a very nice discussion. I shall now invite Dr. Devan Patel, senior most embryologist in Gujarat, to introduce our speaker for the day. Over to you, Dr. Devan.
Yeah, so good evening all. I think today we are having very exciting session on AI. So to start with, just yesterday I was going through the Twitter and where I saw that Chinese company Huawei was showing that with just a hand gesture, you can move file from one device to other device, and it is now the reality. So I think this is really exciting time we are living in. So to introduce, I think our speaker is Dr. Keshav Malhotra. He is a very well-known speaker in this area. Our moderator will be Dr. Rajvi Mehta ma'am, and our discussants will be Sujata ma'am and Sanjay Shukla sir. Over to you, Dr. Keshav.
Sure, let me just share my slides. Very good evening, everyone, and really looking forward to the presentation today and the discussion that will hopefully follow the presentation. We're discussing a very interesting article which was published in Nature, and this is deep learning versus manual morphology-based embryo selection. This is a randomized, double-blind, non-inferiority trial, and hopefully by the end of just knowing about this paper, we'll try and understand how these trials are actually conducted and also try and understand some of the basic strengths, some basic weaknesses of this particular publication. All right. This paper was written by Peter Illingworth and also Professor Gardner, Scott Nelson, Christos Vinitis are some of the key influential people who are present in this study. The study disclosed that it was actually funded by Vitrolife, and some of the authors also received financial support from Vitrolife. So just to give you a background, we know that embryo selection is one of the most important criteria in IVF. We know that we have been following manual morphological based embryo assessments to choose embryos for transfer. This manual morphology based assessment basically, I think a lot of us still follow the Gardner criteria, even though the consensus criteria is out there. But most of us would follow the expansion grade till hatch blastocyst, grade the inner cell mass according to how it looks into A, B, and C categories, similarly for the trophectoderm, and then decide which embryo to transfer or not. Now, when they were actually presenting, or when Gardner was actually discussing this criteria, there were certain students. One of them I've actually mentioned is Angus Tran, who was actually present for one of his presentations. And he actually decided that he can use this data set from time-lapse incubators and then utilize it for a deep learning platform in order to get a predictive tool for fetal heart rate. And that is how IDA version 1, which we are going to discuss in this paper, was actually developed. This AI system was called IV first, and then it was acquired by Vitrolife, and then it came to be known as IDA score. The version that we're discussing today is version 1, but we also now have version 2, which is currently being utilized in the EmbryoScope Plus as well as the EmbryoScope systems. Just to give you a brief overview of the IDA score itself, the IDA score utilizes the image sequences that are generated by time-lapse incubators and then basically analyzes it within its black box. We really don't know what it's analyzing. It's inside that black box, which we all wonder how these technologies work. But then it is predicting whether that embryo is going to result in a clinical pregnancy, and a clinical pregnancy here is defined as fetal heart rate along with the beta HCG value. Now, do notice here that it does not utilize morphokinetic data or any annotations that the embryologist would put into the system. And it also does not utilize any clinical data. So patient age, male partner's age, the kind of factor of infertility that they have, all of those factors are also not involved while calculating the final score for the given embryo. It just analyzes images, and followed by this analysis, it will give a score of the embryo between 1 to 9.9, 1 being the worst, 9.9 being the best. Now, this study was, like I mentioned earlier, is a randomized double-blind non-inferiority trial. We'll discuss these points a little later. This was conducted across 14 IVF clinics in Australia and Europe. In Europe, it was in Sweden, Denmark, etc. And basically, the study design included women who are under the age of 42 and at least had 2 early blastocysts on day 5 to be incorporated into the trial. Exclusion criteria for them were egg donation cycles, PGT cycles, and any additional interventions that were done for these women. Once they were included into the trial, then the randomization was done. So they actually incorporated about 1,066 participants, and they were randomized into the study group, which had 533, and the control group, which had 533. But because there were some patients who did not really follow the protocol of the study, ultimately they ended up with 1,002 participants being incorporated into the study, 500 in the study group and 502 in the control group. The study arm was basically the embryos that used IDA score for the embryo selection, and it was a single blastocyst transfer for these patients. Both fresh cycles and frozen cycles were considered in the study. In the control arm, again, it was standardized morphological assessment, but they also utilized another ranking system that Gardner developed, which we'll discuss a little bit later. The study endpoints, the primary endpoint for the study was the clinical pregnancy rate, like I mentioned. And according to them, this included intrauterine gestation with fetal heartbeat. Now, why was this the primary endpoint? Because IDA version 1, when it was being developed by Angus Tran in the previous paper that I mentioned, the endpoint used to develop this system was fetal heartbeat and not live birth. And that's why they incorporated this as the primary endpoint. But they also considered positive HCG rate, ongoing pregnancy rate, as well as live birth rate as their secondary endpoints. For the stats, the non-inferiority margin that they wanted to consider was 5%. Now, there was a rank prioritization which was utilized for the morphological evaluation as well. Now, we know that there is a selection criteria for blastocysts that we also generally use in our laboratories. Whenever we are looking at a day 5 blastocyst, we're looking at the expansion grade, we're looking at the inner cell mass quality and the trophectoderm quality. So here, according to them, the highest priority was given to embryos which exhibited an expansion grade of 4, 5, or 6. And obviously, AA blastocysts were considered or prioritized for transfer. So top priority embryos in this particular study were 5AA or 6AA on day 5. Second priority was 4AA, 5AB, 5BA, or 5BB. And the lower priority embryos were 4AB, 4BA, 4BB, or lower. Day 6 embryos were generally not considered for this particular study. Now, when you look at the IDA score correlations with the embryologist, the AA graded embryos actually had an excellent correlation with the IDA score, and it was above 8, or actually it was above 8.9 in most of these embryos. Okay, coming to the statistical framework for this particular study. Now, like I mentioned, they utilized a non-inferiority margin for this study, and this was set at 5% for clinical pregnancy rate. Now, this means that the study aimed to show that IDA score performance was no worse than 5% below the control group. So let's say, for hypothetical example, if the control group was performing at, let's say, 38%, which is, or 35.4%, which was the power that was incorporated for the study, they expected that IDA score will at least perform better than 30% in the study. So that's how they wanted to assess. They wanted to assess whether this technology is at least comparing itself to embryology, morphological selection or not. Then the second thing that we want to understand here is the power of the study. So they utilized a 90% probability here. And because their expected clinical pregnancy rate for the control arm was 35.4, they then calculated the power to come up to 992 patients. And the sample size that they required per group was 494 in the study group versus 520 in the control group. And they exceeded this number based on the participants that they had within this study. Mind you, the study started in January of 2020 and went on till September of 2023 when they reported the live birth rates. And this was done during the pandemic, so we can understand how challenging it must have been for them to actually do this particular study and recruit these number of patients for this randomized control trial. The primary outcome analysis was the clinical pregnancy rate. It was noted here that the IDA score group or the study group had a clinical pregnancy rate of 46.5%. The control group had a clinical pregnancy rate of 48.2%. Now here the control group performed better, but because this is a non-inferiority study and we had a 5.5% margin, which was not demonstrated in this particular study. It was actually performing similar. Then when you look at the secondary outcomes, it was positive HCG, ongoing pregnancy rate, and live birth rates. And again, there were no statistical significant differences between these groups. They also incorporated another secondary outcome in this particular study, which was time comparison for basically grading of these embryos. Now, whenever we're doing morphological assessments, it does take us time to analyze the quality of the blastocysts. So here in this control group, if you can notice, it took them about 208 seconds to actually evaluate the embryo. When they compared it to the IDA score, it took them 21.3 seconds to evaluate the embryo. And this 21.3 seconds also included the time in which they switched on the software, uploaded the image. So if we actually look at the evaluation time, it was absolutely instant for them and for this particular software. And therefore, they came to a conclusion that almost 10 times reduction was seen in the evaluation time of blastocysts using IDA score. And ultimately, that's something that can increase the efficiency of laboratories world over. They also tried to do subgroup analysis and intention to treat analysis. Again, basically did not show any significance even in these particular groups. Why was this the reason? Why were they unable to show statistical significance? It was solely because that the expected ongoing pregnancy rate in the control group was much higher than anticipated. They actually anticipated 35.4% pregnancy rate, but the control group performed very well and they got 48.2% clinical pregnancy rate within their study. And if they were to observe if they wanted to observe a difference based on the current numbers, they would actually have had to recruit 7,800 participants to try and detect non-inferiority under these particular conditions and the kind of pregnancy rates that they observed within their clinic. Now, it can be questioned why did they get such high results? There is a principle called as the or an effect called as the Hawthorne effect. This basically was described in 1920, and it comes from a series of experiments that were conducted at Hawthorne Works factory in Chicago. And what was found here was that researchers were actually more productive when they are being observed. So this is something that they thought that could also affect outcomes within their particular study. So clinicians and embryologists might perform at their best, strictly adhere to the protocols that have been defined, or they can pay extra attention when they're being monitored, and that's why they're getting good results. The second thing that can affect the outcomes here was the ranking prioritization that was described for this particular study, where they chose really high top quality embryos for transfer, being 5AA, 6AA, etc. on day 5. Now, when they look at the subgroup analysis, I'll just show you the tables instead of the data. So they did a subgroup analysis for women who were aged more than 35, as well as fresh versus frozen transfers. And again, in the age group above 35, there were no differences. But when they looked at fresh versus frozen results, if you look at their primary outcome, which was clinical pregnancy rate, there was a significant difference between these two study groups, where it was 44 versus 61% um, in the frozen group, where the IDA score did not perform as per expected. Huge difference of about almost 20% was observed in the frozen group. The fresh group, there were no differences. So here, again, they were unable. There are two different talks on YouTube on this particular paper. One is by Professor Gardner himself, and the other one is by Peter Illingworth, both the primary authors of this particular publications. I'd want all of you to go back and see their explanation. But they did postulate that they don't know why they're seeing such big of a difference using IDA score in the frozen group, where they know for a fact that IDA score has performed well in frozen cycles when they were not included in the study. So we don't know the answer to this particular question as of now. It was also seen that when the AI agreed with the embryologist, or when the embryologist agreed with the AI on the basis of which embryo to transfer, the clinical pregnancy rate is slightly higher, not significantly higher, but slightly higher, 48.3% versus 44.7%. This is telling us that instead of AI replacing us, AI in conjunction with the decision making of the embryologist actually gives the best outcomes. And also they did look at the different Gardner grading systems and compared whether the AI was agreeing to it or disagreeing. Again, not much of a statistical significance was seen here as well. Now, non-inferiority basically means that these trials are not trying to prove that the new method is better, but they're actually trying to tell that it's not worse. Right. So this is a new system that is being developed, especially for these new add-ons that are coming up into the market. It is very important for us to understand that these studies have to be powered really well, because like it was mentioned in this particular study itself, when the difference was so small between the two different groups, then we actually would require so many patients, almost 7,800 patients for a randomized control trial. It becomes absolutely impossible for clinics to actually do this. And this has been mentioned by both of the speakers themselves, that by the time that they recruited numbers for this particular study, version 2 of the IDA score was already developed and was already being used by clinics. So it is up to us now to try and find out when to establish clinical significance versus statistical significance, especially when the technology is advancing so rapidly. Key findings of the study, clinical pregnancy rates were similar, but did not meet non-inferiority. Deep learning did significantly reduce the evaluation time. What can be the implications of this? It does imply that AI can improve work efficiency, but does not say that AI performs better or worse than embryologists. High pregnancy rates were observed in the control group, again telling us that it was probably not adequately powered. Certain limitations, all of the embryos were cultured in an embryoscope. Not all clinics will have access to this. Did not assess live birth or time to pregnancy as their primary objectives. And when you look at future directions here, obviously we require larger trials. And we also want to consider the ethical considerations and transparency in AI models. To sum it up, this is the world's first randomized controlled trial for deep learning for embryo selection. The fetal heart outcome was used because IDA-score was developed to predict fetal heart. And how it was developed was that the deep learning black box was given data of the images as well as fetal heart positive or negative. There was no intervention by the embryologist. No other data was provided by the embryologist. So it's like a pure deep learning model that was created just to predict which embryo image sequence can result in a live birth, can result in a fetal heart positive or not. It does tell us that IDA-score performs 10 times faster. And it also tells us that making RCTs for these technologies and keeping pace with the innovation right now is becoming very challenging. What we need larger multicentric trials with long-term outcomes. We need explainable AI. This is again a newer concept that's coming up where instead of the black box just deciding what embryo to transfer, it then tells you how it has chosen that embryo or how the embryo has been selected, so that we also start to learn about different parameters that can affect our outcomes. And we need to establish more robust ethical and regulatory frameworks, especially for AI models. I took the liberty of putting in some potential questions, and maybe I can discuss this and then we can open the floor for discussion. So why was a non-inferiority design chosen instead of a superiority design? Again, here they wanted to demonstrate that this new method is not worse than an embryologist and can be used as a supportive tool for embryologists and not to replace embryologists. So that was the motive behind this particular decision. Why was the margin 5%? Now, this is a clinically acceptable difference in most of the studies that are published in these good journals. They also utilize intention to treat analysis. Now, this basically preserves the integrity of randomization itself. So even if there were patients who were opting out of the study or were not following the protocol to the T, they were still incorporated in the data analysis itself, thus telling us that it's a more realistic measure, which we might see in our own clinics as well. Primary outcome did not show any non-inferiority. Does this mean that AI is ineffective? Now, this does not mean that AI is ineffective. We need larger trials for it, but it does, especially that one particular subgroup analysis where the embryologist was in conjunction with AI and it was showing a trend towards better pregnancy rates for patients. It does tell us that both of us, embryologists as well as AI, can work synergistically to give the best outcomes. Was there any statistical difference when AI recommendations match selections? I've already answered yes, it was better. And can AI reduce the workload? It has been postulated that the use of AI selections, especially this one, can reduce the workload or can reduce evaluation of embryos by almost 10 times. I found this particular publication, which is a commentary or which is in response to this particular paper. This tells us the inconvenient reality of AI-assisted embryo selection in IVF. This is not open access, sadly. So I was unable to read this particular paper, but I found a commentary based on this particular paper, which the few points that it mentions, I'll just tell you. So here, machine learning evaluated embryos led to successful pregnancies in 46% of the patients versus 48% in human evaluated ones. Now, when you look at this particular data, you can spin this in several ways. And this is what was done in most of the blogs that followed the previous publication, where it can be said that machine learning is just as good as human judgment, or it can be said as machine learning was not better than human judgment. The headline actually depends on us and how we want to spin or how we want to define the narrative for such particular publications. Machine learning took significantly less time to evaluate embryos. Now here, yes, we understand that efficiency is being improved, but there was no discussion on how expensive the technology is, because it does utilize time-lapse incubators, which are significantly more costly to buy and even more costly to operate as compared to embryologists. And this prompts us to question whether a reduction in labor expenses and an increase in laboratory efficiency justifies the additional cost, which is relative to time-lapse incubators. And does it say that all of us we are supposed to buy time-lapse incubators or not? We don't know. And therefore, whenever such technologies are being assessed, a cost-effective effectiveness analysis should also be incorporated, because we don't know how these publications are going to be like perceived by the public, and then who is accountable for the patients undergoing potential exploitation by profit-driven technologies. So with this, I'd like to end my presentation here, and we can open the floor for discussion.
So thank you, Keshav, for very smoothly articulating and explaining the entire paper. And especially with a lot of statistics, people tend to get confused, as often biologists are not always as good in statistics and we get floored. One thing the paper surely brings about, that it gives confidence that whatever we embryologists are doing, we are on the right track. So I think that explains the answer to your first question on why not a superiority kind of a study. At least it's proving that we're doing equally well. There were some interesting things that came out. One is that, always we have these highly cliched discussions in meetings, will AI replace embryologists, etc. So this says so. You also discuss something about the Hawthorne effect. So maybe we can discuss somebody observing over you. So, whether you have cameras or recordings when embryologists are working, whether they would create more pressure or actually make you more efficient. It's something by the by, but it's something very interesting on the behavioral patterns that they have studied. Also about something interesting is why they didn't find the same effect in the frozen embryos, where actually the embryologists were better. And you very aptly put about the cost factor. Is it worth the time? Or whether it would bring about more standardization, especially if there are large chains of clinics where you have, variety of experiences of embryologists. So I now hand it over to Dr. Sanjay and Dr. Sujata to take over the discussions, and the audience is free to put in their questions also in the chat. Yeah, over to you. So I think Keshav himself have put. So maybe we can start with the non-inferiority and how confident are you all to get into AI?
Thank you,
Sujata,
Dr.
you were the
Rajvi.
one who was so keen on this paper. So I'll start with you.
No, thank you very much, Dr. Rajbi, and thank you, Dr. Keshav. See, when you read a paper, right, like sometimes it becomes very difficult to kind of comprehend all the points. We would just pick and choose few things, but then like a presentation like yours, the way you have actually done it, I think it makes it very, very easy for people to understand the concept and then kind of, like absorb the take-home messages. Now, the first take-home message I would like to take from this particular presentation was, at present, okay, we cannot completely rely on AI to do the embryo morphology, to grade embryos. The reason I'm saying this is, though I was impressed that it takes much lesser time than an embryologist, but if you just look at the agreement between AI and the embryologist, there is a big difference, isn't it? And sometimes I feel scoring a blastocyst, and especially Gardner's criteria, you actually should not have that much of difference. Unless, like say, you cannot have the big difference between an AA and a BB. So I don't know, Keshav, if you can just, I don't know, can play that particular slide where we talked about the agreement between the embryologist and the machine. I thought that was a bit too less.
1 second, I'll just put it.
So, Dr. Sujata, what I believe,
Mhm.
and whatever, you know,
Yes,
I
this
have,
one.
yeah, so I have, some sort of analogy. So we can compare this AI to a human being developing. Maybe it's still in infancy, so an infant needs directions from its parents, its environment, its relatives, everyone to make that judgment. By the time it reaches, adolescence, it starts making its own decisions then. But still, he or she needs guidance from the elders. So probably this particular AI model is not in infancy, but maybe in adolescence. So it still needs guidance. And once it becomes an adult, maybe we might be discussing the superiority, you know, studies, not the inferiority.
So just look at this. I don't know, both Keshav and Dr. Sanjay can comment, even Dr. Rajvi. See, a 4AA embryo, okay, agreement is only 38% or 38,
36.
36%. I'm just surprised because, like I thought an AA embryo, how can people not agree on that? I understand when there is AB, because, this B is a bit tricky because you are always thinking whether it is B or A, or it can be C also. But when you are saying AA, I don't know why, and see the disagreement,
So
27.
what was happening here was that they had multiple good quality blastocysts for transfer. And when like what if they had a 4AA, there were times where the embryologist might choose a 5AA or a 6AA or a 5AB or a 6AB, etc., instead of a 4AA for transfer. Similarly, in the AI group, if the embryologist was choosing a 5AA, because AI-based softwares are only assessing day 5 embryos, they might score a 4AA better. And that's where this disagreement was happening between labs, between technologies, etc.
See, we have noticed that too in our labs when we were trialing a lot of this, AI systems. The first thing which puts off the embryologist is like when they give a very good grade, which usually is, like not the case. And, embryologists always want to be a bit cautious because the moment they say that it's 5AA and 4AA, if it does not result in implantation, there will be questions. So I have noticed that they always want to kind of give it, one score lower. Okay. And in spite of that, the AI would give a score which is totally deviated from whatever their this thing is, and it really puts them off. And and they do not want to use it. So this is something which I had practically faced uh, amongst our embryologists.
But then
And
once we have data to suggest that
mhm.
AI performs comparatively well as compared to embryologists, then this issue would actually get solved in bigger corporate chains where there are multiple embryologists handling decision making. So I definitely feel that technology here would benefit a group like yours.
Yes, because like standardization is important, and then some kind of a see, when you have an image, anybody can assess. Like me sitting in some other place can also assess that. AI would increase the accuracy, but like it will also help in kind of making our this thing uniform. So definitely that way it's okay. But then the initial reaction is this.
But
And second thing,
also have to consider the kind of AI technology that we're using, because there are systems out there that will just correlate or tell you whether this corresponds to a 4A blastocyst or not. This particular system looked at sequence of images and then predicted fetal heart rate.
Mhm.
Don't have systems out there which have looked at live birth rate and then giving us a score. So it's important for us to understand this as well before we incorporate these technologies in our clinics.
True, very true. And then another thing where I find this would be useful is analyzing large data sets. And here I just want to give an example. The other day in one of the meetings, somebody asked me, like when you have degeneration in the embryo in a blastocyst and when few TE cells are flat, what are the chances of that embryo to implant? And I said, chances would definitely be lower. But then they said, can you give a percentage? Now, to satisfy them, I just gave a percentage. But then I was kind of thinking to myself, if at all the percentage, whatever I'm saying, has to be accurate, I should remember at least 100 cases where I had a similar picture, where there was degeneration, where the trophectoderm was flat and the age of the patient was this much, the sperm parameter was this much. At least certain basic criteria should have been same. But then as a human being, it would be very, very difficult, isn't it, to remember that. So that is where I feel that like huge data sets analysis can be done by this AI and machine learning. And that actually can help us to give some predictive power.
Absolutely.
What, Dr. Sanjay, you want to comment something on that?
I totally agree with you. And in fact, I was also wondering, I do not know whether that information is in the supplementary part of this paper, because I could not read it completely. But it would have been even better if they have provided like the age and other criteria, or the age-wise prognostic factors in relation to this predictions. So maybe because we all know that there are certain conditions which hamper the success rate, and even if we have very good blastocysts, maybe 5AA blastocysts, still we do not get the expected results. So those data, I think it is missing in this. But as you rightly said, that if we have larger data set and then we have the comparisons with other conditions also, probably it may be more useful.
They did do a subgroup analysis where they looked at women who were above 35 only.
Okay.
And they tried to look at the clinical pregnancy rate outcomes for that particular advanced maternal age group. And it was 37.3% versus 39% in the control group. So the study group with IDA score had 37.3% clinical pregnancy rate, and the control group with morphological assessment had 39% clinical pregnancy rate.
So more or less similar.
Yeah, and these were like 90 patients each.
Actually, I'm reminded of one this thing by our Birol Aydin, where he was saying like they had attached some kind of a prism or something to the microscope to make the images three-dimensional. So I know this IDA score is basically from a time-lapse machine, but then I do not know. See, if you are talking about our scenario, right, like the currently available AI softwares which we use, it just looks at a picture. So, definitely the accuracy would be compromised there. And unless the machine is able to look at it in a three-dimensional way, the way embryologist looks at an embryo, I think it's no match, isn't it?
Absolutely.
I also agree with this, and I always wonder because there are AI-based systems out there that look at morphokinetic data, that look at videos, that look at static images. We are now getting new systems that are looking at how like hyperspectral imaging being incorporated into embryonic assessment. And how these systems are actually comparing with each other, nobody really knows. So we can't really differentiate which is the best for our patients, which is the one that we should choose, because they're all out there. It gets really confusing at times.
But Keshav, I'm very optimistic in this direction because there are people who are working to create worldwide neural network with different technologies, and if they all the servers, they are connected with different data sets, and the super computer sort of thing that analyzes that much data, probably we would reach to a better position from here now. And in future, there may be not just one software or one machine learning or deep learning, but it's the combination of all that can give us a better predictions than what we have today.
Absolutely.
Because once I was also invited to participate in one of the incoming project, and that includes the anonymous sharing of your patient's data, not just the embryology data, but also clinical data. And you can compare in real time with similar sets of couples anywhere in the world. So those things are very optimistic and very promising to me.
One thing that comes out from this was about the timing. Sujata, you work with a large group of embryologists. What's an average time without annotation to grade a blastocyst?
Seriously, I have
Approximate.
not, definitely it would be, 3 to 4 minutes, depending upon the number. Like if we have some, around 7, 8 blastocysts, I think definitely they would just take, 3 to 4 minutes, definitely.
In
So
this
this is not clear because here they would have done individual because EmbryoScope has individual culture. So they talk about nearly 203 seconds. So that's over 3 minutes per embryo.
Yeah.
That seems quite a long time. I mean, that's not clear whether it is the group. Obviously, it has to be individual.
Yeah, it
See,
has to be individual.
See, that is
Yeah.
what like when Keshav mentioned like large group of embryologists, that definitely is a disadvantage for us because like it is how much ever we try, standardization is very difficult because there are certain things which are highly individual in nature. I have seen embryologists kind of sitting and admiring an embryo, like totally lost the concept of time. Okay, they're just looking at it again and again. And these days we have kind of restricted them to take these pictures because when they see pictures of blastocysts being posted on LinkedIn or social media platforms, they also get tempted when they have a huge number of good blastocysts. So, but then we have kind of said no. But all these things definitely takes time. Looking at a blastocyst, and then sometimes, like you have to document them. So take pictures of them. So I think it takes time.
Yeah.
One thing which
Also, yeah,
uh, I mean, just a little observation, which is very interesting and which is very, I mean, I'm very happy with this, that even in this era, this particular article mentions that almost or maybe more than 40% of the cycles were the conventional IVF cycles, which is very heartening to me.
So I think if you're talking about that, no, Dr. Sanjay, if you just look at that article in Fertility Sterility, where they looked at uh, consistently performing laboratories in US, and then what are the key factors which they are doing, and one of the point which is mentioned is selective use of ICSI.
Exactly.
Okay, so definitely that has an impact.
Yeah, because I do not know what others' experience is, but I have observed, maybe it's an anecdotal thing, but the quality of blastocysts deriving from conventional IVF is far better than the, morphologically. I'm not sure about the, ploidy status, but at least morphologically they look more, beautiful than the ICSI ones.
I was reading a paper a couple of days ago which talked about ploidy rate compared between IVF cycles and ICSI cycles, and they did show a trend of having better euploid rate with IVF embryos as well. I'm forgetting who was the author, but
Yeah, because another day we were discussing, probably Dr. Rajvi was there, that because
yeah.
all our, fancy sperm selection methods are based on the, things which have come from the natural selection. So it is an expected result that if we have better ploidy status with conventional IVF, it is what we expect.
yes.
And that too, we are selecting some part of the natural selection, whether we are doing PIXI or whether we are doing,
Yeah, HBA test.
microfluidics or all those things. We are just doing a part of it. So, okay, there are some questions coming in from the audience, and one of them is from Nibhash Kumar. He's saying, how do you explain the difference? Why did IDA score not help in the case of the cryopreserved cycle?
I think Keshav has partly answered that.
Actually, the authors also did not know why it did not perform well in this particular study, because it does perform consistently like comparable
in the fresh.
in not just fresh in the patients who are undergoing frozen cycles but were not incorporated in the study. It was giving similar results, but just for this study there was almost a 11% or 12% difference between results. That's something that is probably having a larger data set would help us in explaining uh, what the possible cause for this would be. I don't have an answer for this.
Dr. Sujata, do you have any wild guess?
See, the thing is, like vitrification kind of changes things inside an embryo, right? So definitely, I think they might have to feed in a lot of data on vitrified embryos to come to a conclusion on that. Because, like it is not what we see it superficially. There could be a lot of cellular changes which are happening inside there, which actually, like the system might be able to identify, but then since I don't know how much of training they have done with vitrified embryos, so I could
Keshav, did they do this on the vitrified warmed embryos, the scoring?
No, I'm just reading the explanation here, which is in the paper. They did not do it after warming. The embryos were scored before,
Yeah,
were ranked, and then based on that rank, it was decided which embryo to thaw, which embryo to utilize in the next cycle. Their reasoning here is that in the freeze-all cycles, because the control group would have probably had more number of embryos to choose from, and then based on morphology again they chose a better embryo, and that's how it resulted in a better outcome. But they are speculating that this was purely by chance and not something that negatively affects the use of IDA score.
Also, how much weightage would you give their comment about being observed? It sounds very, whether it's something which would actually be comfortable. I mean, as an embryologist, as a junior, but if somebody's constantly watching over you, the kind of reasoning that they have given, the Hawthorne effect. I
That's
found it a little interesting, but a little difficult to comprehend.
It is a documented effect which has been seen in a lot of trials,
Industries.
not industrial trials, and that's what they are like they're comparing us again with that itself, because when we know that there is a randomized control trial that's happening within our own clinic, we might be positively motivated to perform better in order to get good quality outcomes for the clinic and for the trial itself. And this could be a bias for the study. So, I
So
mean, it's an
Keshav,
interesting.
another thing what I actually is, like found it very intriguing was this concept of statistical significance versus clinical significance. This is something I think which we all have to be aware of in any study for that matter. Forget about AI.
Yeah.
When we are talking about any study, okay, you get statistically significant result, but then what happens to the clinical this thing? Sometimes, like even if you can get one extra patient pregnant, it might be worth clinically. But then if you are just kind of saying this technology is not good because I'm not getting a statistically significant change in the outcome.
That's what, ma'am, all of the current discussions like are pertaining to this particular point itself, because most of these add-ons are unable to achieve statistical significance. But because of this thought process and because of our intention to help the patient, these technologies are being utilized. The caution here is that most clinics are actually charging for use of these add-ons, and that's where we have to then draw a line.
Yes.
We can help our patients, but we then should not be making profits out of these technologies.
Exactly. And then I think that is again, it justifies the intention of looking at a technology and finding out whether it is causing any harm. So this non-inferiority.
But
No,
again,
because
yes.
Yeah.
Other thing that's happening is that by the time we are able to recruit good quality data for a randomized control trial, a newer technology is already crept into practice. So again, waiting for collecting so much data is also becoming a challenging situation for clinicians and embryologists now.
do we have any more questions from the
Yeah,
audience?
or anybody wants to opine or give some opinion or discuss, please feel free. You can chat, or you can even unmute yourself and.
Yeah, so I do have one question for Dr. Keshav. In this paper, it is mentioned like deep learning was done. So the number of patients were mentioned were 1,000. But have they mentioned how many number of embryos they have used for deep learning?
No, so the deep learning model was actually based on the previous paper and not this particular paper. So this one, this is the data that they actually utilize. The paper that I'm mentioning here, this is a 2009 publication by Angus Tran and his group. And I'm not sure how many embryos were utilized for generating this data, but obviously it was.
Keshav, it mentions here. So they included 1,15,832 embryos to train them.
To train. Yeah.
It's a huge number of embryos.
Yeah.
Right. Thank you.
And all of these embryos had fetal heart rate, positive or negative, as the outcome. So it's a good quality.
Yeah, I mean, they
Yeah.
mentioned that 14,644 were transferred with known outcomes. So almost 15,000 embryos.
So just to include, My question is like, there are certain patients who are having, just for example, like first 1,000 embryos are coming from maybe 50 patients, maybe 20 each, but other remaining 1,000 is coming from maybe 500 patients, two each. So can there be a bias if, for the deep learning?
I think so. I had actually two slides on bias. I can just put those slides for you. One second. I'd not incorporated these because of like I wanted to stick to time. But whenever we are trying to generate data with AI, there are a few biases that are associated. First is our data bias itself, which most of these AI models actually have, because most of the data is coming from a specific region and it may not represent global diversity. I think that's one that answers your question. Second thing is that there is a selection bias because we cannot generate prospective data for 1 lakh embryos. It's impossible. It has to be retrospective. And again, when we are using retrospective data, there is a slight bias, even with experienced embryologists, that they will give in better data which favors embryos which are selected by experienced embryologists or which resulted in better outcomes. It's not truly like a randomized data that would be provided. And again, clinical and practical implications of this, basically it will not result in consistent outcomes world over. And again, we're completely worried about standardization.
So obviously we need more data and more time
Yeah.
to reach to some conclusion.
So just to give some take-home messages for people who are there from the paper and from the current scenario here, AI definitely is a promising technology, but as of now, it may not offer any extra benefit over morphological screening. But that shouldn't deter us from even trying, because, like one, as the technology grows, we also need to get used to it, like the kind of data input and all that. And we can't wait for the final, the best format to arrive, and then we start to learn how to use it. And with the current systems which are available in India, I think a lot depends upon the picture quality, what you're giving, because your accuracy would depend mostly on the kind of pictures you are giving. And then you have to actually kind of correlate it or double check with the embryologist morphological selection, because still there is an issue of this agreement and disagreement between the system and the embryologist. This is from me. Keshav and Dr. Sanjay can add to this, and then I think we can close.
Yeah, just one point from my side to supplement what you have said, that unless we give enough time and enough data to the machine to learn it its own, that's what the deep learning is. So unless we use it, we allow it to analyze our data and images, it cannot reach to that extent where we can totally trust on this. So we need to at least incorporate it in our day-to-day practices. May not be offered as a trusted partner at the moment, but yes, unless we allow it to analyze the data, it cannot be used. So yes, it is a promising thing, and as we grow with the AI itself, of course the machine learning and deep learning will grow, and then we will be able to predict more precisely than what we are doing at the moment.
Absolutely. Points from me would be, I think we should really celebrate this trial, because it's one of the first multicentric randomized controlled trials, registered trials on deep learning and comparing deep learning with morphological selection. And I think it gives us enough perspective to design future trials, design future studies, see what needs to be assessed and what we can actually bypass. A word of caution here for people who are using AI-based technologies. Most of them are actually based on day 5 images and day 5 selection. And there are times where we will get blastocysts on day 6, which if we analyze with AI might not give the best score or a comparative score as compared to a day 5 embryo. So we still have to build up on data for day 6 blastocysts as well. So that's pretty much it. There are two good videos out there on YouTube on this particular paper itself. One is by Professor Gardner. If you're interested in this, please go see his presentation. The other one was by Peter Illingworth. This is on Vitrolife's platform. So if you're interested, you can check their like what they actually felt when they did the study and how they've described it.
So we come to the end of this session. The science of AI is moving so fast. So like Keshav said, by the time they set up a prospective study, the technology has moved. But that shows that, yes, AI is a thing of the future, and the two greatest benefits would be time. It does save a lot of time. And second benefit would be the standardization, which with humans and new people coming in or individual variabilities during one's functioning would be taken care of. So we just have to keep a watch. Just because it's a new technology, we don't need to jump in. And the question just comes in all the time is when something new comes in, who pays for it? And somebody has to pay for it. So that's something which we'll never really find an answer. It's your own conscious and how you can manage in your situations. But yes, it's surely a technology for the future. So I thank everybody who attended. I thank Keshav for his very succinct presentation, and Sujata and Dr. Sanjay and Dr. Sujata for leading the discussion. And thank you all for joining in. So our next ACE online program will be very soon. So this time we'll be having some conversations with eminent embryologists and scientists in the future. So thank you so much and keep in touch. Bye-bye.
Thank you.
Bye-bye.
Thank you, Keshav.
Thank you.
Bye-bye. Thank you, Dr. Rajvi. Thank you all. Bye.
Thanks, Devan.
Thank you,
Bye.
ma'am. Thank you,
Bye.
ma'am.
Bye-bye.
Bye.
Bye-bye. Bye.